to have our very first, and we're all on this, which is great, shape and away from each other, very important. Yeah, so this is our first of a series that we've had running for many years now. This is the Center for Computational Science visiting lecture series. We have our lecturer today, Wonmuk Hwang. And what we're going to be doing over the next few weeks and months is have several visitors coming. In 2 weeks' time, there'll be another one of these, which will be Ibn Shan, from
former Tcell.
former Tcell.
Yes.
Okay.
Yes.
I didn't realize it was a former Tcell.
Yeah.
Okay.
You can talk about that.
You can talk about that later. Okay. And so the idea is that we will have today's visitor, and I believe also John will be here for a week or so.
He could be.
Yeah. Okay. Great. So the idea here is to have people come, leading scientists from around the world, visit us for a week. Okay. So Wonmuk is actually in the office back there. You can use your 5 key to get into the main area, but you can't get into this area with a 5. You need a 7 to do that. We go into this today.
Yes.
And so you can go and talk with him throughout the entire week. You're going to be here, right?
Yep.
And so the idea is if you learn something that you're interested in this morning or this afternoon rather, go and see what the connections are. We can talk to him and take advantage of the fact that we are paying to have him come and join us here. Okay. And so you are now going to introduce him.
Okay, yeah, that's it.
Fantastic.
All right. So I won't take too much time from Wonmuk anymore. So just a brief introduction. So Wonmuk actually is an alum of BU. He received his PhD from the physics department, working on condensed matter theory. And so maybe he'll say a few words about exactly what he worked on as a student. And then he went to MIT to work with Roger Kahn, so a very well-known mechanical engineering, that we're biomedical engineer.
Yeah.
And so anyway, so very interesting area. And so that's when Wonmuk started to look at biomolecules, right? So since then Wonmuk has been looking at the broad ranges of problems. I guess I know him mainly through the work of molecular motors, right? And so that's something that we have been interested in as well. But as you can see from his title, right? And so we're going to go to pretty large length scales as well, right? And so all the way to the brain. And so I look forward to, the talk and the visit again. I encourage you guys to go and talk to Wonmuk. And so he clearly has broad interest and a lot of different expertise. And as I sort of mentioned to some of my group members, right, in addition to science, right, and so Wonmuk has also other interesting, I guess, expertise. You can ask him about how to run marathon effectively in Texas weather, and also you can talk to him about classic Korean music, as I understand it. So anyway, so without further ado, please.
Well, thanks a lot for invitation, and it's a great pleasure to be back again. Yeah, as Chang introduced, I was a physics PhD student doing condensed matter theory. So at the time, I had no idea about biomolecules, and I didn't know what amino acids were and so on. So, but condensed matter theory is about these complex systems and self-organization and so on. I mean, those all reflect what biology does. So there was actually a connection deep inside. And I was assigned to this office, and this morning, David said there's only roof. So I took a picture from the window. Actually, it looks pretty good.
It's very futuristic.
Yeah. Yeah, so I have an interest in many different systems, but the underlying theme is kind of similar, as you will see. So this is just brief overview of my perspective. I started from physics, but now, there is almost no barrier across these disciplines. And because I'm in an engineering department, many people actually don't know or understand what I'm doing. Yeah, that's true. So Yeah, the thing is, the biological systems life, there is no single discipline that can like fit here. So we are just trying to understand this from different perspectives. And these disciplines are built mainly as like, human social activities. So when we study this, we have to break all the barriers. Yeah, and again, because I'm in the engineering, I was telling the coach from Robert Oppenheimer that society supports science for two disturbingly different reasons. One is useful, the other is ennobling. And yeah, I would agree that about this usefulness and ennobling. I don't know if they have to be disturbingly different. Yeah. Yeah, so what aspect I'm interested is mostly like structural and mechanical. So I mean, this view is from David Bowe's book on cell mechanics, like describing the cellular structures as assemblies of one-dimensional filaments enclosed in compartments, membranes, and cross-linked and moved by motors, which are like zero-dimensional objects. Yeah, so these are the range of systems I'm interested. Yeah, I will not going to go with like technical details. I'll just mainly focus on what the problems are and what computer simulations and analysis can do about these questions. Yeah, MD simulation, I guess, need no introduction here, right? I'll just skip. Yeah, so let's start with the DNA. I mean, this is like scales about DNA. I mean, every cell we have has DNA about 2 meters long. If you stretch them out, yeah, there are like base pairs. Each base pair is like 4 angstroms long, and there are like 10 to the 9 base pairs. So if you multiply, it's like 2 meters. And each DNA is about 2 nanometers in diameter. So if you just get the volume, it's about 10% of the nucleus volume. I mean, it's not impossible, but considering that DNA is the most negatively charged molecule in our body, this is going to be tough. I mean, the packing. So DNA is packed with histones and so on into other like chromosomes that you probably have seen. But the problem is, say if you get a signal, then the signal tells us to transcribe certain genes. Then you have to find where the genes are located, and you have to unfold them and read the sequence and get the mRNA and so on. So this local flexibility matters. And if you go to a bigger perspective, yeah, when we do polymer studies, we can try, describe polymer as a line. That's a great achievement done by Flory and Duzan. But in biofilaments, it's not just that like contour length related behaviors, but actually the sequence matters. So where DNA is more flexible and less flexible dictates how they can be transcribed and so on. So the problem here is how to describe this DNA as an elastic rod that reflect the sequence-dependent behavior. And the task was to find the proper order parameters for describing DNA mechanics. So the simulation studies was like this. These are 10 possible DNA base pairs, AATT, ACGT, and so on and so forth. Then you like 100 nanosecond simulation and study the motion of one base pair relative to the previous one. And this is the like sprinkle plot for this axial vector. And you can see the orange dots, which tells how this base pair moves relative to this. Then it is an ellipsoid, ellipse. So there is a major bending direction and a minor bending direction. It's like if you have a ruler, it's easy to bend in thinner way than the transverse way. So from DNA, you can find this major and minor axis. Then you can apply the equipartition theorem to get bending stiffness. Then there is a study well described in Landau and Lifshitz statistical mechanics book about the persistence length of this anisotropic rod. Then you can study which base pair bends in which direction, easier or harder. And if you have that information, then you can build a coarse-grained model of DNA that reflects the sequence. So this is the flexible AT-AT, and this is stiffer AG-AG. So this can be used to build up larger scale behavior of DNA that reflect the sequence. Yeah, so let's just move to the other part about the solvation of DNA or biomolecules in general. And yeah, an impressive aspect of this water is described in Israel Lachivli's paper that most scientists were accustomed to thinking of interactions and forces between particles. If you have two particles, you think how they interact with long range or like non-bounded directions, but actually the surrounding solvent medium is as important in terms of mediating their interactions. So I've been developing so-called the Solvation map of this water. And in this case, we studied the methylated DNA. So these two spheres, methyl groups in cytosines, and when our cells try to silence certain genes, they methylate certain regions. And also cancer cells have irregular methylation pattern and so on. So there's a interest about the methylation. And these blobs are the high density water shells around DNA. And there are these characteristic double Ds. And if you turn it, now we are going to the backside, the minor group. In minor group, there are deeper water molecules, so-called the water spine that can be seen in the crystal structure study. Yeah.
What's the color?
The color is just a radial distance from the center.
So the reddish is closer.
Yeah, right. Yeah, so we've been developing this to understand interaction between molecular surfaces, like hydrophobic or hydrophilic and so on. And currently what we are doing is try to get the solvation energy based on this hydration map. Yeah, there are these Poisson-Boltzmann method and other types of solvation theories, continuum-based. And if there are also the simulation-based methods, and in the simulation-based methods so far, people fix the DNA or the protein. But if you fix, then actually the surface hydration changes greatly. So you have to keep them moving. And since we can calculate the density map, we can calculate the solvation energy basically based on the density of space in a sense. Yeah, KBT log the density over the bulk density. So if this is higher than the bulk, that means water likes that place. Lower than the bulk, water doesn't like that place. I mean, there are a lot of technical details of doing this for a simulation trajectory, which we are currently developing. Yeah, so we moved from filament to solvation, and now let's use that to understand the immune system. I'm just quickly switching topics. So there are also amazing facts about this Earth. I mean, if you go look at it from distance, actually the Earth is a microbial planet, and we are just kind of exceptions. So if you take 1 gram of soil, there are 10 to the 11 microbes. And how many grams of soil the Earth has is enormous. And if you just take our own body, 90% of our body cells are non-human. They are microbes. 10 times more than our own. If you take your palm, there are over 150 species, and in our gut, there are more than 1,000 species of microbes. So when we say V or I mean, it's hard to, yeah, we are just kind of excitations in this sea of microbes. Yeah. So that tells us, I mean, we have to be interacting with them in order to survive. And most of these microbes are obviously harmless, or even they are needed for survival. But some become pathogens, and sometimes harmless microbes can become pathogen depending on our body condition. So this immune system or immune surveillance is constant. It's not just we think about immune response only when we are sick. It's just a basic process in the microbiome. So here are like 2-page summary of the adaptive and innate immune system. So we have this innate immunity where we have genetically encoded receptors that recognize, say, bacterial polysaccharides. So if we get infection, we recognize those polysaccharides, then we can fight. But there are these viruses and so on that mutate, so we have to deal with that. That is the adaptive immunity. I mean, the range of these diversities tend to be like 10 or even more. If you think about the exposure to microbes throughout our life. So we have to have that many receptors. And obviously we only have about billion base pairs, 10 to the 9. So we cannot get just more than 10 to the 9. If we just genetically encode all the possible receptors. So what our body does is we have gene segments and we assemble them in different combinations and also introduce mutations to generate diverse receptors. And there are B and T cells, the main adaptive immune cells. So these cells generate the antibody, which we hear a lot these days due to the coronavirus. And T cell is less well known to the public. So when we talk about antibody, I mean, here's Dr. Fauci from NIAID holding this model of coronavirus. So the spike protein is this, and antibodies recognize mostly this outer surface. So when there's a variation, the mutations, antibodies don't work well. The T cell receptors or T cells can recognize the inside. And inside is important for the stability of this protein. So they are less varied because if the virus mutates inside, the stability of the spike becomes like less, then they cannot survive them. So most of the mutations are on the outside. So how does a T cell recognize the inside? That's the inner part. The next slide. And so what our cells do is we like process or chop off these proteins and present them to the surface by the molecule called the major histocompatibility molecule, MHC. And they are named MHC because they were the molecules found to be like causing the graft rejection in the early days. So almost all the cells in our body have like hundreds of thousands of these MHC molecules on the surface. And almost all of them will hold peptides from our own protein. I mean, we constantly process these proteins, and when we degrade the proteins, some of the proteins end up on the surface, telling them, oh, this cell is ours. So we have self, but when there are infections, some of the proteins are now displayed on the surface, and we have to distinguish between self and the foreign. That's the main challenge here. And that's what the T cell does. And because the protein is broken down, inner part components are here, which is how the T cells can recognize the inner part of this protein. Yeah, if you chop this out, there are peptides generated from the inside. That's what we can recognize. So that's it. In that sense, Tcell is actually very important. And when we get vaccinated, it's not just antibody, but there are T cells that can recognize, which is how our vaccines work against these variants. Yeah, so here's the structure of the T cell receptor. And this is the dimeric T cell receptor. And this is roughly the surface of the T cell. And this is the surface of any antigen presenting cell, almost any cell. And this is the MHC structure. So you have alpha and beta 2 microglobulin, and this is the peptide held by these 2 alpha helices. So the T cell receptor has to tell whether this is us or invader. But this peptide is only 8 to 10 amino acids long. And the T cell receptor has to be able to pick out of hundreds of thousands molecules. Almost all of them have cell molecules. If there are a few copies on the surface of the cell, T cell can recognize, okay, that this cell is infected. So if this is infected, 1 in maybe 100,000, there is a foreign peptide. And the binding affinity is low, greater than micromolar, typically. So from biochemistry, are you all chemists? Here, sort of, yeah. So micromolar, greater than micromolar affinity is called undruggable. It's so weak. And antibodies typically work in the nanomolar range, pretty strong. So how does this conundrum arise? Weak binding, but very sensitive. Yeah, so this is the main test for TCR. And recently, my collaborators, Matt Lang at Vanderbilt and people from Harvard Medical School, they were beginning to see one big contribution of this sensitivity, which is the catchpoint or mechanosensor aspect of TCR. Oh, this doesn't move. So yeah, when we apply force to adhesion, yeah, like sticky tape, if you apply, you can detach them. Exponential decay of bond lifetime. But there are some biological contexts that increase the bond lifetime with force to some extent. And only when the force becomes great, they decay. So this is called catchpoint. You strengthen the bond by applying force. I mean, yeah, this is basically due to conformational change of this protein that becomes like better sticking when there's a force. And in this single molecule assay, the surface has the peptide MHC molecule, and here is the alpha beta TCR, and that's pulled by this optical trap. And they measure the bond lifetime. And as you can see, VSBA is vesicular stomatitis virus octapeptide. And that's the agonist that needs to be recognized in this system. And this bond lifetime is longer. And L4 is a mutant from valine to leucine at position 4 in this peptide. The catch bond is weaker. And this is the Sendai virus, nonapeptide, and it shows a slip bond behavior, irrelevant. So this is highly dependent on what peptide is there. And moreover, they made a mutation of delta FG. So this is the FG loop. This is like a protruding elbow in this beta chain. And when they deleted it, they saw the suppression of catch bond. And the reason why they studied the FG loop is this is like vertebrate adaptation. So if there are organisms below jawed vertebrate, so there are organisms that are vertebrate but have no jaws, they don't have this T-cell receptor. And yeah, from jawed vertebrate up, there is an FG loop, and mammals actually is the longest. So there's a clearly evolutionary relation. So this seems to be enhancing the catchpoint behavior. So my study was how this actually happens from structural point of view. So I built different structures, systems, like the complex, the regular complex, and without this peptide MHC, and without the action loop to see how the behavior differs, and without the so-called constant domain, but a peptide MHC, and so on. I mean, these are not all of the systems I studied. There are actually more. And the simulation goes like this. Yeah, you hold the ends with harmonic springs and just run it for like 500 nanoseconds. And this pink cloud is just a center of mass trajectory of this peptide. And as you can see, the force vector instantaneously fluctuates. I mean, there's a thermal aspect too. So this is a low extension, high extension. So when people study this adhesion or bonds between like complexes, usually what people do is just apply high force called steered molecular dynamics to lead to dissociation of the complex within, say, 100 nanoseconds or so. But if you apply very high load, then the unbinding pathway will be different from the natural. So what I was trying to do was to apply force and see how they respond. That can be done within 500 nanoseconds. Because if you consider propagation of sounds, which is basically phonon, from here to there, it takes about 10 to 20 nanoseconds. So 500 nanoseconds was long enough to see conformation of, say, equilibrium.
Quick question on this.
Yeah.
So how do I see that the bond is strengthening here? Is there structural reorganization? It's not obvious what it is.
Right. So that'll come now.
Okay.
Yeah. Okay. Yeah. So I measured many things. So first thing was the occupancy heat map. So you have the crystal structure. There are amino acid contacts of, if you run the simulation, this contacts form and break during the simulation. So I measured the instantaneous occupancy with this rolling average, and actually plotted this occupancy heat map with a different occupancy cutoffs. So this is reflects a particular set of cutoffs. So what you can see is the low extension, there are many reds, meaning that there are low occupancy. High extension, there are more blues, meaning that they contact more strongly at high extension. And no C domain, if you delete this lower part, actually the contacts increase, which means there is some allosteric effect from here to that domain too. Yeah, so I measured like the Hamming distance from the beginning and changed this occupancy, and analyze the distribution, spatial distribution of these contacts within the interface, and all of them point that high extension or high load is better than low extension. Yeah, I mean, you cannot calculate the binding free energy, I mean, with this method, but you can tell which one is stronger or less strong. Yeah, then how do you connect the interfacial dynamics to the conformational motion? That's the key. So I did this principal component analysis using machine learning, I guess, and what's shown are the first three modes, which I call the twist and the scissor and flap. So as you can see, these purple spheres, they are the CDR complementarity determining region loops that contact this peptide. So they are the most important in this receptor. And what we are seeing is as they move, their arrangement changes. So if you do not suppress this motion, the interface is bound to be unstable. Yeah, and I also studied like the context between B alpha, B beta, B alpha, C alpha, B beta, C beta, and also actually look and found the contexts are not like evenly distributed. First of all, between these two that we saw the motion, the number of contexts differ the most depending on the system that you're studying. And between C alpha and C beta, the contact count is like 30 or so. So this is the most robust based. And between the B alpha and C alpha, yeah, contacts only go up to like 4. So these are weak, strong. And there are some contacts depending on the simulation condition and so on. So the main thing is the distribution of contacts are uneven. And when we go to that picture, in this case, you need to satisfy 3-body contact, which is non-trivial. So if there's a domain motion here, you can actually control how well they fit. Yeah, so that's the idea. And the C module is an agitator actually, because without the C, you can get better context. So the C module was there from evolution or whatever to destabilize interface without force. So skipping all the other analysis, here's the lesson I found. So without the load, these domains are not like perfectly fit. I mean, it's topologically closed structure, so you cannot just deform them and just easily fit them. And also, if there's a mismatch in that type, the same thing happens. And when there's a load, then there are these contacts between the MHEC to these B domains to pull them to stabilize their contacts. So these domains don't move. Then if the peptide is the right fit, then you can maintain the interface. So here, the mismatch seems to be there from the basic design of this Tcell receptor. And the catch bond is achieved by stabilizing this domain motion. And before this work, people thought the right peptide will be the load-bearing thing, but actually it's not. I mean, there are only a couple of contacts between the peptide and this, whereas there are much more here. So peptide is more like is a teeth of a key that filters the right fit, but once the fit is good, then the load-bearing contacts are different. So currently, what we are doing is we study this for different types of Tcell receptors, and the pre-Tcell receptor. This is a simulation of the pre-TCR, and I use basically the same method that I use for the DNA to study the relative motion between this and that and that, and so on and so forth. And I mean, if you think about making receptors as sensitive as this, you can make this as like a one-shot development. So actually, when the Tcell receptors develop, this beta chain is first expressed without the alpha chain, and their contacts are first screened. Then after a certain group of contacts are selected, then alpha chain is expressed to finally get the mature TCR. So the problem here is, okay, when you don't have the alpha chain, the contact between this beta and PMHEC is not going to be used in the end. It's going to be only a transient contact. Then what do you mean by the right or wrong contact here? So those are the questions that need to be addressed. And there are experiments, but the atomic picture, I guess, can only be obtained from simulation. Yeah, so I'll be quick in what's the time here? Oh, this time is good. Yeah, move to the motor protein. And I was fascinated with this motor world. And this was from a review paper I wrote with Methylation. And the main output of this review paper is this picture, actually. Yeah, 5 nanometer scale block. So they are actually the same scale. So these are the motors, RNA polymerase and ribosome, and yeah, other DNA modifying enzymes, myosin 5 and kinesin. And these are the tracks where these motors walk on. And yeah, the understanding develops from the experiment first. So this is made by Ron Bale's group at UC San Francisco. And a lot of it is imagination. Some negligible. So there are kinesins that walk backwards. So they actually switch this domain between the two kinesins. They actually switch the direction out. Although the motility is quite impaired if you just switch this. So yeah, it's not like a point object that does what. I mean, we have to understand basically the atomic details here. So I did a bit of simulation using Anton actually. And yeah, without explaining what the simulation was, here, this is a supplementary movie explaining the concept developed. So it's less imaginary, but still a lot of it is imaginary, but a lot of it is based on simulation. So yeah, the front head explores the binding site by kind of diffusive search. And the rear head, it has a bound ATP. And note that they have to make steps. So the interface has to be marginally stable. Actually, there are water molecules going in and out. So it's a wet fit. Yeah. Yeah, then we pay attention to the ATP, and these are the switch domains. And the simulation showed that these switch domains are not just rigid, they actually move quite a bit. But they still move within the density of electron dense cryo-EMFs. So there are these mobile switch domains, and the conformation of ATP in the free state and the bound state. I mean, this is more based on the crystal structure. There is a torsion developed. When the torsion doesn't break the bond, but I think it'll help with the attack by water. Chang may have more to say about this. Yeah, anyway, so ATP hydrolysis make this weaker bound, which we don't know exactly. And ADP release in the front end. In the simulation, you can see the ADP pulled out, but from here to there, it's just a visual demonstration. So yeah, now apostate, no nucleotide, and then this had releases PI that was seen by simulation. And okay, the ejection is helped by this moving loop. The loop is pulling out the phosphate. So in the ADP state, the binding becomes weaker, then this can detach. So this has kind of moves around, which is not well understood right now.
Did you just make sure that you're still sharing the screen? I don't think you will.
Okay, let's see.
No, not yet.
Oh, sorry.
Now you are. Okay,
Yeah.
good. Thank you.
Sorry.
No worries.
Yeah, then in the front end, there is ATP binding going on. And the thing is, ATP, this is adenine part, which is like hydrophobic part. This is the phosphate, triphosphate, which is highly charged than hydrophilic. So if you just put this near kinesin, this part wants to go in. So there is a electrostatic guidance for this negatively charged part. And also what we will see is that these mobile domains actually help with their orientation and binding. Yeah, this is the actual simulation that they are basically tossed by these three. And yeah, the binding, this, I just made it happen. Yeah, so ATP binds to the front head. Then we have the neck linker I mentioned. And how does that move forward? There is a small N-terminal strand called cover strand, and they form a beta sheet. And the beta sheet has a forward bias to push this forward. So this is how one step is done in kinesin. So yeah, there are other questions about this conformational change and so on. But what's missing big here is how they move when they are like unbound, or how do they diffuse? I mean, those questions are, I guess, the least understood. So which is what I'm studying right now. And those questions also relate to the family dependent behavior. Yeah.
The simulations do not have a load on them.
There's no load here. Yeah. I
So
studied.
presumably the load has a huge effect, right? It should drag along a very large thing, right? So
Oh, yeah.
This is the
Yeah.
load-free case. You know how big there's a load. What sort of load would you need to pull it backwards, right?
Oh, yeah. So the load has to be more than, say, 10 piconewtons and so on to stop. I mean, kinesis tolls at 7 piconewtons. So if you apply 7 piconewton, they cannot walk anymore. Yeah, and the most load sensitive phase of this cycle is when they try to make the fourth step. Yeah, if you pull back, yeah. But what I showed was when they form the beta sheet, then they can withstand the load and move forward. If it's a single strand, I mean, there's no way they can stand the load. Yeah. Yeah, and there are other kinesin families that do amazing behaviors, like they diffuse and they depolymerize microtubule. And yeah, they stabilize microtubule and so on and so forth. So there are quite a few questions left in the kinesin family still. So switch our gear for the remainder, like a few minutes. So this is like an offshoot of my biomolecular simulation. The question is, yeah, if you see an image like this, you train them through some machine learning, then you can tell this is the starfish and these are sea anemones. But if you want to study their patterns or like length distribution or motion of these tentacles, that's a different story. And people have studied or generated this models of different structures. Here's a mitotic spindle. I mean, this is an experimental image, and this is Drosophila braid, a neuronal network, and this is, I think, yeah, zebrafish larva braid. So there are these traces, and if you go to the supplementary information, they boast how much effort they spent, like 450 days of uninterrupted human annotation. So yeah, with all this development of machine learning methods and so on, there are tools that assist with this tracing, but this in the end like by hand. And the problem is, okay, so suppose you did this for one system, can it serve as a ground truth? The connectivity is all different. So here the ground truth is, yeah, out of question almost. And I mean, I got into this based on my earlier work. So we studied this collagen network on mica surface in like AFM image. So I was developing like theory about their network formation, then wanted to measure their length. And it wasn't easy, and actually it happened in the end. So these filaments are colored differently, meaning that they are registered in the program individually. So I can measure the orientational distribution. So you can see 2 peaks, -45 and 16.2, that are major. And 73.8 is this. So you can like roughly feel there are less filaments in this direction than that or those directions. Yeah, you can measure this orientation distribution, and you can measure the length distribution in the log scale. And there is a little bit of power law, 2.6, showing that this is like a fractal structure. Then you can build a stochastic model of this structure formation. You start from like empty space, then you create certain filaments at certain rate, and you grow them in certain growth rate, and growth stops when they hit other filaments.
Like great screensaver.
Yeah.
Okay, thanks. Yeah, so this is the simulation, and the nice thing is that you have this experimental data, then you vary your like growth rate and see what is the dimensionless growth rate that can match this distribution. So once you find this growth rate, then you can do the simulation, and it qualitatively matches the experimental structure. Yeah, so this method is further developed, and now I'm doing this microtubule tracking. So these are the microtubules on like 2-dimensional substrate. So there is uneven growth. This is a fast growing ant, and they do something called catastrophe. Then they polymerize it. So this needs to be tracked to get their velocity distribution and so on, or how frequently they do catastrophe. And here, the efforts have been there previously. So Tcell is the program that I developed, which actually traces the manual the closest. And the most advanced so far is the M-track, which you can see there are occasional dropping and a little bit of lagging here. And so just to be fair, I downloaded images from this M-track website and applied their tutorial and applied mine. And actually the ends are better tracked in my method. And this is, yeah. The problem is these are written in MATLAB or Java, so anybody can use these. And right now, this is script-based, so it's just difficult to use. Yeah, but this has a lot of applications. I mean, yeah, although this seems to be very different from simulation, the ideas underneath took from MD simulation. Like we use bees and bonds and these quadrilaterals, the simple objects, and assign the objects to structures. And in this case is zebrafish embryonic brain. So these are the neuroepithelium. So if you just apply the simple binary thresholding, it becomes noisy because of this background. But there are a series of refinement procedures, progressive and error tolerant. And for any given image, we can separate them into different channels and look at different aspects. So the combination becomes this nice surface map with starting from quite noisy image. So actually this biophysical reports, that was the first issue when we just covered the first issue. Yeah, so many interesting things going on. The fun thing is that I like this programming and debugging and so on. So this was, it's amazing, already 10 years ago, my first build cluster that I built. So it was quite exciting. So this was a shot. And I won't mention why this doesn't come from this Korean scholar's name, but So the first simulation was for Martin Corpus, 32 picoseconds, 458 atoms. And with this cluster, I was able to simulate 10K atoms, about 1 nanosecond length. And now the time for building your own cluster is almost gone. We use all this large scale, and yeah, there's we took the picture. Yeah. Anyway, so yeah, Martin mentioned, MD simulation can see the ultimate details of motion of phenomena. That was quite a right statement, I think. But I think you have to know what you are simulating and how to analyze and so on. So these all have been great and exciting developments so far. And I'd like to thank many people. So I'm just showing relatively recent people. And the current active collaborators are Jay Hancho, Martin Kaplis, Matt Lang, Alex Reiners, and Maria Zhang. And my group here, and with all these funding sources and competing resources. And now I'd be happy to take questions.
All right, it was really impressive, breadth of things that we have learned. So open for discussion.
Do you know what the catastrophe is that causes this change?
Oh yeah, that is GTP hydrolysis. So microtubule, I mean, if it does polymerizes in one direction, that means there must be free energy cost. So free energy is supplied by the GTP. So when a tubulin monomer has a GTP bound, it's prone to polymerization. So they polymerize, but as they age, GTP hydrolyzes. So this hydrolysis follows the front. So when it reaches the front, if everything is like in the GDP, they depolymerize. Yeah, so it's the balance between hydrolysis rate and the growth rate. Yeah.
Yeah, I have a question about first of all, thank you for talking. That was really interesting. Question about the recognition of the peptide in the immune system.
Mhm.
Did I understand correctly that the weak binding that it's supposed to detect, like you mentioned, that it's very weak binding.
Yeah.
So that's the mechanism is specifically built for that, right? Because if the force, that's meant to be the weakest force.
Yeah. Yeah. So without force, these are almost indistinguishable. And this 10 to 20 piconewton force is actually the physiological level force.
Yeah.
I mean, when we do this immune surveillance, T-cell moves relative to the antigen presence. They move around. So when they make transient contacts, they move on. That's the pulling force. Yeah, that has been measured, 10 to 15.
But I was just, so the fact that in the presence of force, which I understand is physiologically relevant, is when this thing works best, I think necessitates that the linking, that binding, intermolecular binding to speak. Otherwise, it would denature the molecule itself before.
Right.
Is that a right way to think about it?
Yeah, that's one aspect. And the other aspects, I mean, if they are strong, then what happens is, I mean, these T-cell receptors are sort of randomly generated in our body because we don't know what to expect. So we have these random T-cell receptors, like millions of them in our body. And if they are strong, and suppose some of those are made to recognize the self-peptide and binding is strong, that will lead to like autoimmune response.
Yeah.
So actually, during the development of this T-cell receptor, there are positive and negative selection phases. So positive selection means the T-cell receptor has to have some basal binding strength. So those are initially selected. Then among them, they are screened for negative selection. If they are too strong interacting, they are deleted. So you only have a range of interaction strengths that's weak. That's, I think, how they select weaker than the stronger binding yeah, again, if they are too strong, I mean, yeah, even if they may not unfold, it's going to be difficult. Just like the kinesin walking with wet feet.
Just to know that Marsha is our expert on, let's say, optical tweezers and torturing the molecules and so on.
Nice.
So I'm sure you have a lot to talk about. Any other?
Yeah,
sure, David.
So the fact that you mentioned this several times, the feet are wet.
Yeah.
Why is the wet important?
Well, because when they make detach from the surface, if they are dry, that means it's mostly like hydrophobic interaction. Then it'll be difficult to detach. So the binding has to be something that can be easily broken by some tiny conformational change. And it's better to have water interface and hydrated interface. So if something is wrong, then they can easily detach.
So
Yeah.
the water is electrostatic free.
Or
Reducing the strength.
yeah, if they are a little farther away. When they are in the hydrated case, the distance is within the debye length. So the screening will be difficult in that bound state. But when they are like this, then yes, the screening can play a role. And actually you can control the speed or the run length by changing the salt concentration. Yeah.
Since you have this slide, right, I think you mentioned that, for different organisms, right? So the length of that FG loop is very different, right? And so, have you, explored that and, have a hypothesis or?
Not yet. I mean, first understanding human or mammalian is challenging enough right now. So if you have a very good understanding, then we may move to the other organisms. That's one thing. And the other thing is the availability of experimental data. People mostly work on mice or human.
I guess it makes sense.
Did I actually understand you correctly? So you said vertebrates, but you said vertebrates with jaws?
Yeah, jaws.
Jaws. So what the hell is the connection between having a jaw and doing this and not having a jaw and not doing this?
So I had exactly the same question. So I pondered about that. So jawed vertebrates, I guess, have more, diverse range of food to eat. So they are exposed more organisms. So that's my guess. Yeah. So which is why they need to have like more advanced receptors. And then the other question was, then are there vertebrates without jaws? And I hope the internet works.
Yeah, what was that? What is the example?
Yes. So there.
Oh,
This lampreys.
it looks scary.
Yeah.
Really?
Yeah. Looks like from this alien movie.
Alien
Something
movie.
else.
Yeah. Yeah. So they have mouth without jaw, but they have vertebrae.
Well, the last part of your computation, right? So, essentially refinement of that brain image.
Yeah.
I understood. And so you said that, you were basically applying, let's say, basic elements of MD simulation, right? And so.
Some methodologies.
Right. So, but I'm curious, where are the parameters, right? So what's needed to develop those parameters to make the refinement work?
Yeah, first of all, there is no force field here. It's just a static structure. The idea was to represent this in terms of bees and bonds and so on.
Right.
So if you read about this image processing literature, almost all of them work at the level of pixels. But each pixel doesn't carry information about this structure.
Right.
So you need some sort of source, coarse-grained picture, which kind of inspired how to use this like geometric object assignment. So B here locally represents some features of a group of pixels, like 9 pixels and so on.
Right.
Yeah. Then connectivity and so on and so forth. And yeah, this output is written actually as a term PSF and coordinate files. So then you can visualize using PMD or other.
Right, but I guess what I'm curious about is, I mean, there's representation, right?
Yeah.
But then usually whatever computation you do, I
Yeah.
mean, you have to like either optimize something or maximize something, right? And so what is being, what is the cost function?
Yeah, so those are the kind of refinement. So if you just do this beta chain assignment on a single image, it's rough.
Uh-huh.
But you have these layers of chains, then if you apply, say, the continuity of the surface, because the real structure, the surface won't be this rough. So if you apply the continuity, then it becomes smoother. So it's kind of like applying energy minimization locally on the surface. Yeah, that's one way to look at it. And yeah, the other way is speed, because this image analysis takes a lot of time if you just use Java or Python or MATLAB. But if you write in a C++ and make it actually the command driven, it can be much faster.
But to follow up on this question, right? The parameters of the spring constants for the bonds and various other things that influence the shape of the net, right?
If you
So-
want,
So how robust are the structures you predict for this, or the structures you visualize for this, the choice of those types of parameters?
yeah, so those parameters you have to, I mean, right now it's kind of semi-manual tuning. So you choose how many pixels you want to use for assigning a BDR. I mean, that's usually determined by the length scale of these features. So over what range of length does this change significantly? That gives the range of, say, what do you call it, a bead diameter, for example. And what is the permissible angle between bonds? Yeah, those depend on the individual images. I mean, if the image had the kinks, then we should be doing something different. Yeah.
Cool. Oh, I mean, there are people online still.
I think so.
Any questions from online people?
Yeah, our virtual
We're,
audience.
Yeah, go over there.
Go, the quick thing.
There's John Stroud with it.
Yeah. Hi, John, you have any questions?
The problem is I don't.
Oh, I can't hear you. Yeah, that's right. Somehow we can't hear.
Can you hear us?
I can hear you.
Oh, yeah, they can.
Okay, but we can't.
We can't hear you. Oh, yeah, the problem is, my laptop doesn't have, there's a sound from here.
I mean,
No, there's not.
realistically, you could type out the question, right?
Yeah, if you have question, you can type into the chat box.
Actually, my sound might work. Can you give me the link?
Oh, the link.
The passcode?
Join the meeting. No, the
Uh,
actual meeting.
Oh,
Oh,
Actually, you go to the top of is that little hot,
here?
no? There it is, 965. 965. 0755. 1077. Join. Now I need the password. Just a minute. And the password is 092023. Continue. Okay. Got it. Wi-Fi.
Recording in progress.
Okay. Too many devices. John, do you have a? John, do you have a question? Is John still there? John is still there. Can you hear us, John?
maybe you can.
Yeah. Say something?
Can you hear me?
Yes, we can.
Okay, I was just typing a question, so thanks for a great seminar. I mean, going back to the beginning, we were talking about DNA organization and chromosomes and so on, and I was wondering if you've seen any sort of out of equilibrium dynamics or, glassy sort of behavior in any of those systems.
Yeah, DNA can unfold and so on. And our model is limited within the linear elastic regime. So our model right now doesn't address those transitions and glassy behaviors. And glassy behaviors, I think, are more for the aggregate or the assembly of DNAs rather than individual base pairs.
What
obviously-
are your thoughts on those more sort of large scale, ultra coarse grain models of DNA?
Well, actually, we compared our model with OX DNA, I think from Oxford. They had this coarse-grained model. And in terms of capturing the mechanical behavior and sequence dependent, our model works well because their OX DNA was more for the transition, and I don't think they parameterize their DNA like the classic properties based on the sequence dependent way. So by design, their model is, their model is focused on something else than what we studied. Yeah, so in terms of DNA looping and persistence lengths people measure, our model gets the numbers sort of better.
So you're more of a proponent of sort of bottom up coarse graining, starting with more microscopic models and coarse graining rather than the top down for these types of systems?
Yeah, I would say so, because I think coarse graining has to be based on certain properties that you are interested. So if you do top down, sometimes if the behavior is very regular, then you can capture the underlying dynamics from top down approach. But for a lot of biomolecules, they are very finicky. So you have to understand their atomistic behavior first before building the coarse-grained model. And yeah, in that lines of thought, I am not very fond of generic coarse-grained force field such as Martini. They have, I know that you use Martini, but it has its uses. But say if you want to study like motility of kinesin or aggregation of certain amylogenic proteins and so on, it's better to put the or system dependent parameters in your model. That way you can capture the behavior better, I think.
Yeah, I mean, I think it's a very interesting question. It probably depends a little bit on the question you're asking, right? What type of model can reach that. But I know, there are people doing very good work like on RNA, Giovanni Busi, that are more proponents of a top-down model. Greg Voth, of course, is a proponent of bottom-up. And so anyway, probably depends on the system.
Yeah, and besides, I think bottom-up is more time-consuming in a sense than top-down.
Is more what, sorry?
Time-consuming to develop.
Oh, yeah. Good, thank you.
Thank you.
Right. Thanks, John. Any further questions? Okay, let's thank Wonmuk for a really great talk.
Thank you very much.
Thank you.
David, are you using quantum teleportation to send me that lunch?
We're working on that, John. We're working on that.
Not quite there yet.
Want to try it out?
We have a coarse-grained version of one. It was bottom up, though.
All right.
Have fun,
Take
John.
care. Good to see you. Thanks.
Enjoy.
Bye-bye.
Bye. Okay.